So our next speaker is Mark McPeek, who has graciously agreed to come in and give a talk, which was very spontaneous because May Berenbaum is ill and was not able to come. Mark earned his bachelor's and master's degrees from the University of Kentucky and his PhD from Michigan State University. He is currently David T. McLaughlin Distinguished Professor of Biological Sciences at Dartmouth College, one of the few colleges in the United States that still calls itself a college, and proudly so. Mark's interests are remarkably broad and integrate ecological, biogeographic, and evolutionary approaches to understanding the dynamics, assembly, and structure of biological communities.
Oh, thank you, Jessica. So, as Jessica said, I'm not May Berenbaum. I also have no genetic data to show at the population level, so some of y'all can relax. I was invited to give this talk. I was 30 miles from here on the Long Island Expressway driving here when I got the invitation, so bear with me. And some of y'all have seen this talk before because I didn't have time to print out a new one. So I'm an ecologist, as Jessica said, primarily, and what I've been interested in for a while are ecologists are having a really hard time trying to explain patterns of species diversity. And what I've done in the last 10 years or so is gotten interested in trying to figure out whether there are other kinds of data we can bring to this problem of explaining extant patterns of species diversity. And what I really think is important is that we really have to think about not only how those species interact today and how those interactions affect who can live together and who can't live together, but also how past evolutionary processes in those various interacting species have brought them to have the adaptations they have today, and how those processes may have influenced diversification of those lineages over time. So if you're going to study a problem like this, what you really need is an evolutionary history of them. But most of us work on taxa that do not have good fossil records. So we are sort of stuck with molecular phylogenies to try to reconstruct the history of the past. Now the problem with doing phylogeny, so I study communities today, and we may be able to reconstruct the habitat distributions or some sort of ecological characters that influence how these species interact with the environment they find themselves in. The problem is, if you work with phylogenies, is it's an incomplete picture. So we have to keep reminding ourselves we're not seeing the whole picture of the past, but what we can do is to give us some idea about what speciation, extinction, and adaptation has done in the past to lead us to the species we find today. So that's what I want to talk about. And particularly what I want to talk about is what drives diversification in this talk. So if we take this kind of data, so here we've got a nice phylogeny that I simulated on a computer, assuming a particular speciation rate, and in this case, no extinction. And so we get an overall diversification rate, which is just speciation minus extinction rate, and we get a nice phylogeny out the end. Now we can analyze and try to back calculate and estimate these parameters from this phylogeny by doing the following. So if we start at the base of the tree and we simply sweep across the phylogeny and plot the number of lineages over the history of this clade on a log scale, we get this curve. The slope of this curve is expected to be the speciation rate in this case. So we can, if we just take molecular phylogenies, plot these what are known as lineage through time plots, we can estimate speciation rate from this, according to our standard model of how phylogenies work. We can also calculate a number of statistics that come off of this plot. One of them that people look at in particular is a statistic that Pybus and Harvey have really championed, known as gamma. In this case, when the rate of lineage accumulation is constant through time, we expect gamma to be 0. Another statistic I find very interesting is the distribution of the ages of the species at the tips of the phylogeny. So if we estimate, we just use the external branches as an estimate of how old the species are, and look at this distribution What you expect in this case, again, with speciation rate greater than 0 but no extinctions, is a coefficient of variation among those distributions of 1. So you basically get an exponential distribution for this frequency. Now, how does extinction change this process? So here's a phylogeny I've simulated with a speciation rate here with some extinction, but again holding overall diversification rate the same as what we had in the previous example. In this case now, the slope over most of this lineage through time curve is expected to be speciation rate minus extinction rate. But in the very recent, we get an upturn because extinction is not really important for extant species, so the extinction rate goes away, and this last little upturn estimates the expected speciation rate. So from this plot, we can back calculate speciation and extinction rate. There are a number of studies there's a lot of theory that goes along with this. There's a lot of empirical studies of pulling these things out of the molecular literature and calculating these parameters. In this case, when we see this acceleration where we've got speciation and extinction occurring, what we expect is a gamma statistic that's greater than 0, which implies an acceleration of the rate of lineage diversification. We also expect, in this case, a coefficient of variation for external branch lengths greater than 1, because in this case what we'll end up with is an excess of young species. So we can use these two statistics to actually say something about patterns of diversification that we take out of molecular phylogenies. So what do you see in real phylogenies? There's a number of studies that have looked at collating data out of the literature. And Jackie Brown and I have done this for 245 molecular phylogenies that were all at the species level. So they're basically clades of species where we go back to whatever the author of the paper has put in their clade. Here's the data we get. So here we have a plot of gamma along the x-axis, the coefficient of variation for external branch lengths along the y-axis for 4 major groups: chordates, arthropods, mollusks, and magnoliophytes. So we've got plants and animals here. All 4 of these major groups show the same pattern. Most taxa, so in our data set, 81% of the clades have a gamma distribution less than 0. They also have external branch length CVs less than 1. Which is very surprising. And this is a result that continues to pop up in these independent collations. So we have groups here scattered across animals and plants. Phillimore and Trevor Price have done this same analysis for birds. They basically get the same result. Negative gammas. How can that be? So our standard model for how diversification occurs expects at least positive gamma values. Why are they so negative? Well, the way you get a negative gamma is to actually have a decelerating rate of lineage accumulation. So you end up with a phylogeny that looks like this. Most of the branches are deep in the tree, and you have long external branches. So if we look at this lineage through time plot, we have rapid diversification early in the history of the clade, and then the rate slows greatly. If we look at the distribution of species ages estimated from external branch lengths, what we get is a coefficient of variation much less than 1 because we have an excess of old species in these clades. And this is the pattern that we see in most of the clades we've looked at, and most of the clades other people have looked at. Now, just to give you a little idea about what this data looks like, here are some data from our data set. So this is actually the oldest clade, one of the oldest clades we have in the data set. This is from McKenna and Farrell studies of beetles in the Amazon. They show a really nice rapid diversification over 25 million years, and then it slows down. But even really young clades show the same kind of patterns. And you also see this excess of old species in these taxa. Now, How can we modify this standard model of diversification to explain this pattern? Basically, the only way you can do this is to come up with a model where you have basically some sort of very rapid decline in speciation rate at some higher abundance. So we make effectively the speciation rate diversity dependent. In this case, what I've done is built basically a step function into the speciation rate here with no extinction. And this is the distribution that we get out of simulating phylogenies under this model. So we get negative gammas, and we get coefficients of variation less than 1. Beautiful. The problem is, let's now go to a situation where we hold overall diversification patterns constant across species richness in this clade, but now we have a little higher extinction rate. So now we let extinction into the problem. What happens is this cloud of points shifts to positive gammas, and coefficients of variation greater than 1. And if we kick extinction rate up higher, it gets even worse. So the problem here is that extinction erases the history of diversification in a clade. And once you think about it, it makes a lot of sense. When you drive clades extinct in a phylogeny and you prune branches off the tree, you end up erasing all the history that's deep in the clade, that's deep in the phylogeny. So what we get from this is a prediction that doesn't really fit very well. And it doesn't, I mean, those guys back there are, they shouldn't like this. Because what it implies is speciation rate can be diversity dependent. That's fine. But what it implies is that there's no extinction. Or extinction is completely unimportant in the history of clades. We know extinction happens. So how can we rescue having extinction happen in clades and still reconcile it with this data? Because this is the standard model that people assume and that people are analyzing, looking at molecular phylogenies and trying to estimate diversification rates from them. What I want to propose to you is maybe what's missing from this whole exercise is the ecology in which this diversification happens. And maybe if we build in an ecological process into this and we allow these clades to diversify according to the ecological principles that they're interacting with one another, maybe we can get back and understand this and have reasonable expectations about the results that we get out of it. So let's think about for a minute the dynamics of speciation and extinction in an ecological context. How does ecology influence speciation and extinction? Well, let's first think about speciation. Right? Speciation is accomplished by a huge number of different modes of generating reproductive isolation. Right? If we look in any textbook of evolution, there is a list of speciation mechanisms as long as your arm to generate reproductive isolation among things. Some of those are critically tied to the ecology of those lineages. For example, ecological speciation, as its name implies, is reproductive isolation that comes as the byproduct of ecological differentiation. So immediately the speciation process is creating species that have very different ecologies from one another. We all know lots of examples of this. Jonathan's going to talk about a really good group tomorrow morning, so I won't need to belabor that. But other modes of speciation don't generate a lot of ecological differentiation among the taxa at the end of the speciation process. For example, lots of ways of sexual selection generate changes in characters where females and males make breeding decisions with one another, but the characters that change to create those differences between the species don't affect the other types of interactions that they're involved in. They don't affect how they deal with predators, how they deal with resources, how they deal with mutualists, other than pollinators, how they deal with diseases. Lots of characters may change and you just get new species out the other end. I work on insects, right? If you're going to look at insects, you do a lot with genitalia. When the genitalia of my insects change, they're new species, but it doesn't affect how fast they can run away from a predator. Now, the degree of ecological differentiation can also influence dynamics of extinction, right? So extinction, we, as an ecologist, typically think of extinction as not having a lot of temporal dynamics. If a species is going to go extinct, it'll go extinct and we'll be done with it. But extinction may take a very long time. Some taxa and some processes that are driving extinction may knock lineages out really quickly. Other lineages that are being driven extinct, it may take a very long time to complete that extinction process. Now, what are some of the things that may determine these rates? Well, so I like to explain this in the context of a boxing example, right? So think about a boxing match, right? So think about the time it takes for a boxing match to be completed as basically the time to extinction, right? If the two competitors are very poorly matched with one another, such that one competitor is much better in whatever competitive interaction they're involved in than the other one, time to competitive exclusion is very short, right? This fight lasted, what, 45 seconds? So unequal competitors lead to short fights. So if you have very different ecological capabilities of these species, extinction may be a very rapid process. On the other hand, if the competitors are very well matched, the fight takes a long time. It takes a very long time. And the competition can be very intense. So these competitors may be fighting and fighting and fighting, and when you go in and look at the strength of competition, it's extremely strong the whole time. But because the differences in competitive ability are so small, it takes the winner, well, I can't remember which fight this was, but it takes the winner a very long time to beat the loser. And then we get the same results if you look at competitions in models and you look at it in the laboratory, competing species together with one another. If the difference in competitive ability is very small, the time to extinction is very long. If the difference in competitive ability is very big, the winner wins very quickly. So let's build a model and see if we can rescue these patterns that we see in molecular phylogeny. So what I've done is I've been playing with a model for a while now where I build what we call a meta-community, which is just a collection of patches that are connected by dispersal. So in this model, what we're going to assume is each patch lies at some point along some environmental gradient. Pick your favorite environmental gradient. This can be multidimensional. It all still works the same. Here I've just color-coded the positions of each patch along this environmental gradient. And we're just going to assume that the ecological differences among these patches are random draws along this environmental gradient. Now, let's assume that each species has some competitive ability along this gradient, such that there's some optimal point for a species along the gradient. And that occurs at random with respect to where the patches lie along this gradient. Right? So for this species, this species actually has its best competitive ability in the yellow patch. And away from that point on the gradient, its competitive ability falls away. Another species may have an optima at a different point along the gradient. And so in the yellow patch, this species would beat that one rather quickly because their competitive abilities are very different. In this patch, in the lavender patch, this species would win because it has a higher competitive ability. If we think about speciation, speciation is basically just creating species where you take the progenitor species and make a new species at some random point along this patch. And this is how we're going to play this game. Now you can make the derived species very different, or you can make the derived species very similar to the species that it's derived from. So just to run through the way we're going to do this, so this is all simulations that we've run. I'm going to assume here that speciation rate is actually constant. So there's a constant per lineage speciation rate in this model. So everything has to do with extinction dynamics here, instead of changing the speciation rate with diversity. The degree of ecological differentiation is created by basically just drawing a random number from a Gaussian distribution with an expectation of 0, but what I do is manipulate the standard, the spread of that Gaussian distribution. extinctions are driven purely by ecological interactions here. So the ecological dynamics in this model come into the extinction rate. And what we're going to assume here is just basically plain old vanilla Locke-Volterra competitive dynamics among the species in all these patches. Now, I've played around with a number of variables in this model, but what I'm going to focus on here is the degree of ecological differentiation at the time of speciation. All these other things that we can vary about the model really don't have a big effect on what I'm going to talk about here for the next few minutes. And basically, as we run these simulations, I'm just basically going to keep track of what the phylogeny of the species are in this system to try to reconstruct a molecular, what would be comparable to a molecular phylogeny for the species, and keep a fossil record of them. Today, I'm just going to talk about the phylogenetics issues. So what do you get out of this? So I'm an ecologist, so I'm interested in the number of species that you have in the system. This axis here is the spread of that Gaussian distribution that I'm assuming. So over here, new species are very different potentially from their progenitor. At this end of this axis, new species are all very similar to their progenitor. And what you see if you run this for 500,000 iterations and just count the number of species after 500,000 generations is a U-shaped distribution of number of species along this gradient of change in the ecology of species at the time of speciation. So you can think of over here as ecological speciation and over here as sexual selection. Now, the really interesting thing about this is that over here, so if you play the game and you ask how many of these species that are in the system at any particular time are actually transients through the system, how many are being driven extinct? So you can ask that question by basically just turning off speciation after 500,000 generations and just wait until you don't have any more species going extinct. What you see when you do that is this curve. So at this end of the gradient, where you have primarily ecological speciation happening, all these species have their own place in the system. So nobody goes extinct when you turn speciation off. As you decrease the ecological differentiation you generate at the time of speciation, you get more transient species in the system. So that when by the time you're over here, most of the species are slowly being driven extinct. Over here, species are being driven extinct really rapidly if they're going to be driven extinct. So the difference in this line is basically how much species richness is inflated by this transient dynamics in the system. Ecologically, over here what you end up with are species that are very well spaced along this environmental gradient. So it basically looks like a limiting similarity plot you'd see in a community ecology textbook. In the middle, you get some separation of species, but here, because clades are not making very different species, you have a limitation of the clades to explore the full extent of the gradient. And by the time you get over here, what you end up with is just a pile of species that are very, very similar to one another, all in the same spot. So how can we test this? Well, let's look at the phylogenies we get out of this. So we'll make the same plot for different positions along this gradient. So with ecological species, with conditions associated with ecological speciation, when new species are very different from their progenitors, we end up with phylogenies out of those simulations where gamma is negative and the coefficient of variation is less than 1. Just what you expect. As we make new species more similar, so we move to the left on that other figure along that axis, this cloud of points shifts, such that by the time we get to what's equivalent to sexual selection, we end up with positive gammas and coefficients of variation much greater than 1. So if we look at the ecological dynamics of this system and forget about the phylogenies for a minute, in this end of the gradient, what we see is an accumulation of species in the system at a pretty constant rate until this clade effectively fills up the gradient. And then after this point, the only thing that happens is species replacements. So if you think about what species replacements are going to do for a phylogeny, if all the species replacements occur along one clade, you get no branching. You just get replacement, and that branch just gets longer and longer and longer. If you do have species replacements, you may put splits into the tree. But here we still have lots and lots of extinction occurring. But in this niche filling phase, that's deep in the phylogeny. And then later on, we have this, what I call the species replacement phase, which is after that curve is bent over. Right? So if we go back to this real data, that component seems to be consistent with this part of the accumulation in ecological space. And this is the species replacement phase where we just are getting species replacements. But those extinction events are not being written into the history of that phylogeny. Most of them. But in this model, extinctions are still happening. And in fact, extinction is what's driving all this. Now, if we look at the other end of the gradient with sexual selection, what you see is basically an exponential accumulation of lineages in these simulations. And what we get out of these is a nice plot that we expect, where we have a speciation minus extinction rate, and then at the very recent, just see speciation in these clades. Right? So let's go back to the real data just to wrap up here. So what we see in lots and lots of these phylogenies is something that looks like this. So this is the lineage accumulation plot for the clade that has the very largest positive gamma value. But if you look at this lineage through time plot, what you see is there is this slowing deep in the history of the clade, and then this upturn in the very, very recent. So there seems to be signals of both ecological speciation and non-ecological speciation modes in the history of this clade. And you see this pattern over and over out here. Most of these clades show this kind of double, niche filling at the bottom, species replacement, and then a very big upturn in the very recent. Well, what clade is this? So the overall one for this value is just about 5. If we cut this off here, we get a value of gamma of negative 1.37. Well, it turns out I know a little bit about that clade because that's the clade I work on. The history of, this is Enolagma, they're damselflies. There are 38 North American species. Ecological speciation is a very prominent mode of speciation in this clade. There have been lots and lots of ecological shifts to create new species in this clade, but those are the minority of speciation events that have occurred. Most of the speciation events are associated with breeding. So changes in sexual parts that females and males use to identify one another to species. And most of the history of this upturn here is speciation in this clade, even though a number of these speciation events in this most recent upturn is are also due to ecological factors. So I think one of the take-home messages from this is that no clade speciates by just one mode. Every clade really has a conglomeration of various mechanisms of speciation happening all at the same time. And what we see in the phylogeny is really just the relative frequencies of those different modes. So that Enolagma is way out here because sexual selection seems to be dominant. I think the lizards Jonathan's lizards, I think, are somewhere over here. The primary mode of speciation in Jonathan's lizards are ecological speciation, with other modes of speciation probably being much less frequent in that clade. So that I think building ecology back into this system and thinking about the ecological processes that are driving speciation and extinction allow us to rescue this sort of blatantly false assumption that or a conclusion we'd have to make that extinction is not happening to explain the molecular data we have. So one of the things that this sort of exercise has taught me is that sometimes it's not the data that's wrong. Sometimes it's the model we're assuming to analyze that data that's what's wrong. And that we always have to question ourselves about, well, sometimes we may have to just take the data at face value and rethink how we're conceptualizing the processes we're building into this. Now, there's also, I just want to put one more plug in for this because my guys don't really do a lot of ecological speciation, is that there may be some bias in these data sets as well. Because what we've taken out of the literature are molecular phylogenies that are based on sequence data. So really rapidly and recently radiating clades, you don't get a lot of diversification in sequences. And so what we are missing from this data set are things like Hawaiian Drosophila and African lake cichlids and silver swords and all of those taxa, because systematists use other markers to try to generate molecular phylogenies from this, and we restricted ourselves to sequence data. So I think there's probably more taxa over here than the data would actually suggest, but that's just me because mine happens to be way over here. So what I want to leave you with is that, when we're studying, for me, a biological community, a lot of what we're trying to explain are the same metrics of nature, right? There are a lot of us that are trying to explain species diversity. There are a lot of us that are trying to explain patterns of genetic variation among populations. But a lot of times what we do is we're sort of like, the wise men, the blind wise men grabbing onto different parts of an elephant. We only look at the kind of data that we use. So I'm a community ecologist, so we usually do field experiments, and we tend to think, well, the Earth is the way it is today, and it's always been that way. But I think what we need to do is to start trying to reconcile across different disciplines for all of us who are looking at the same parameters and trying to reconcile, well, the way you think about this doesn't make sense to the way I think about it. And if we're looking at the same parameter of nature, they've got to make sense because we're looking at the same thing. So I think I'll leave it there. Thank you.